So my name is Liza Makowski, and I'm a PI here at University of Tennessee Health Science Center. And first of all, I'm really thankful for this opportunity to present, and very thankful that that was Boston's first talk. And so I'm really proud of him. And he introduced this mouse model that my lab has started using, and I'll explain how that's happened. So I came from UNC Chapel Hill, and my family moved to Memphis in 2017. And just a little bit about Memphis. We heard a little bit about social determinants of health this morning. So this map shows the cancer mortality in the Mid-South. And so where is Memphis? It is right here at the tail end of Tennessee. And so along with the Appalachian Mountains, we see that this Mid-South region has some of the worst cancer mortality in the United States. By the darker the red, the worse mortality. So there's a real opportunity here to come study and change cancer. And so let's get deeper into that. So Memphis here again in this middle of this slide is in this region. And if you notice, the closest NCI designated cancer centers for adults or medical centers are noted here. And some of them are ranging, hundreds of miles away. And Memphis is right in the middle between St. Louis, Little Rock, Nashville, Birmingham, Alabama, and Jackson. So this area is ripe for an NCI designated cancer center, which is why my husband and I moved to Memphis, because this is really the only complete basic biomedical, clinical, behavioral, social science, and health services area where we could build this NCI designation. And the dot you do see here, so that's St. Jude's, but that's a pediatric child hospital. And when those kids turn 18, they need advanced care. So I was really excited to move to Memphis. And a majority of my work has focused on breast cancer. So I'll introduce this a little bit. So we know that there's different subtypes of breast cancer, and it's clinically diagnosed, or in the lab, we could look at RNA sequencing by looking at hormone receptors, such as estrogen receptor, progesterone receptor, or HER2. And while least prevalent, the most aggressive cancer is triple negative. And so these patients have the worst outcome and very few treatment options because most of the drugs target estrogen receptor, for example, like tamoxifen, or newer drugs. So when I came here, our goal was to try to work with the team at UTHSC to identify novel genetic modifiers of breast cancer traits by using some of the tools here, such as unbiased systematic quantification of BC severity and heritability across strains. So I'll talk a little bit about this, but the later speakers in the session, also from my lab, will go into further detail. So you just heard the BXD mouse introduced by Boston. And this is one of the best and largest characterized genetic reference population, an excellent review by David Ashbrook, the session chair. And while started about 15 years ago, it's been expanded to over 150 strains at UTHSC and deeply phenotyped with lots of data available in the Gene Network database. And Boston has also introduced this genetic mouse model of breast cancer. And as mentioned, it's a progressive model, which makes it good to study both kind of the onset and progression. So even though it's transgenically driven, I'll show you that we do see differences. And this progression is well defined. So we see early proliferation. So this is a normal duct where in the center, this is where milk would flow in the mammary gland. And you see the luminal cells and the basal cells around the duct. And that's where either luminal or basal-like breast cancer can come from. And this is a model to also answer your question, where it's more basal-like. And we see this progression from atypical hyperplasia to a pre-neoplastic lesion called DCIS to the invasive actual carcinoma, or the tumor, over time. And so this mouse also has been around for decades, and it's been well studied. My group has studied a lot of obesity, which you'll hear about in the next talk. Other groups have studied exercise, but it's been most studied for pharmacological approaches. But it is a single genetic background, which is why we're here today. So as Boston mentioned, the mice get either breast cancer or prostate cancer. And there had been no previous genetically engineered mouse models of cancer in the BXD. So we were excited to work with Rob Williams and the group here for the Center for Integrative Translational Genomics. We got some pilot mice and we started to cross. And when we first were working with Lulu and Mel McCarthy and Chasey Chapman, we said, which mice should we use? Should we choose a specific strain or should we just go random? And so we randomly got some females. And so as Boston said, we crossed the males that were transgenic for the cancer into the BXD strains. And we were really surprised to find some variability in the tumor phenotypes. And again, in these hybrids, they're spontaneous, so they're genetically driven to form tumors, and all the females get cancer. And I'll at the end, I'll just mention some studies on prostate cancer. And so again, these are really complex and lots of breeding, huge breedings. And really, Casey has helped us a lot with colony management. So I want to shout out to her. We spent a lot of time characterizing these outcomes. Hang on, let me hide this. Where is it? Thank you. Okay, so characterizing these mice. So click back in. So female mice have large litters, so they actually have multiple mammary glands, and you basically have to give the mouse a massage all over their body in the front to feel all the mammary glands. And once you can palpate them, and my lab can do this when they're the size of a front of a pen, we start noting that as latency. So when did they get their first tumor? That's latency. And then we follow them over time. And we've had lots of undergrads involved, and this has been a great project to bring in a lot of students. We've also collaborated with Dr. Robert Reed, a pathologist, and some other students, and you'll hear almost last in the session from Jeremiah on RNA-seq. And so in looking at some of the data, when we first were getting our pilot data back and looking at, this is a Kaplan-Meier plot looking at basically survival or which mice are tumor-free, we see over time, like Boston showed you, the FVB mice were right in the middle, and we did see some really early onset tumors and later onset tumors when we follow them over time. And so this is just really early pilot data, and you'll see some of these strains, like 161 or 44, some of these strains, like 161, they had their tumors come on very early at 15 weeks, whereas others were coming on weeks later, which in this mouse is pretty significant, showing that we did have this variability. So we had either early onset or later onset cancers. So even though they're transgenically driven by knocking down RB and P53, we see variation in onset. And so I won't get into all the data, but we hope that soon this will be in bioRxiv and then available on Gene Network. We also worked with veterinary pathologists and others to look at other measures of cancer markers. And for example, we looked at some differences in, to answer your question, to look at some gene expression. So we looked at, by RNA-seq, looked at some of the estrogen receptors, progesterone receptors, and even androgen receptor. And as Boston noted, or maybe I'll remind everyone, we had a couple of strains that did not develop tumors, which was really exciting for us. We're in the process of repeating this. And Samson will talk a little bit more about that. But in the mice that did develop tumors, we saw lots of variability. Like, for example, some of these had very relatively higher expression of androgen receptor compared to others. And one of our colleagues is really interested in androgen receptor and has drugs actually in clinical trials on androgen receptor. And reassuring to us was that the parent strain, their tumors were triple negative. And so then we generated all this data over years. We said, David, Rob, Lulu, we need help. And so we entered the data into Gene Network and saw that a lot of these traits were indeed significantly heritable. Like, for example, I'm noting the multiplicity of tumors, and you'll hear a bit later from Samson about tumor latency. And so here's again looking at tumor multiplicity. As Boston pointed out, some of our mice strains in the BXD-BC crosses had very few tumors per mouse, 1, 2, and then some of them had tons of tumors per mouse. And then again, we had these mice that did not develop tumors, and we just looked at their mammary glands. And so the data that we currently have is about 27 strains with 2 that never developed tumors. We're currently adding 28 additional strains. So we hope that we can have really rich and robust QTLs. For example, I'm going to show you one QTL, and you'll hear some more later from Samson. We see this QTL present on chromosome 16. And in working with David again, we can take the synthetic regions from the mouse in certain chromosomes or certain hits of interest, and then correlate those or look at human data and GWAS data. And you'll hear from Samson in a little bit about a gene, for example, that we found that had a significant QTL, was significantly regulated in patient tumors and associated with survival. So I will let Samson talk more about that. And then once we get these candidates, like some of you today presented knocked out or knockout or gain of function models, we will also then turn to either in vitro or mouse models to then validate these candidate genes with classic cancer biology approaches. So proliferation, invasion, which is cell motility, or migration, again, cell motility. And so to end, I really wanted to call out my postdoc, Laura Sipe. She is my former postdoc. She's currently an assistant professor at University of Mary Washington, and she got a grant for this from the AACR Foundation, American Association for Cancer Research, that funded some of her time with me. And just to reiterate, Laura did this incredible amount of work kind of as a side project on top of a bariatric surgery project, which you'll actually hear about from another trainee of mine, I think Wednesday. And so I'd just like to end by saying, despite being funded, thanks to all of Laura's preliminary data, we have an RO1 and a UO1 to look at BXD breast cancer mice. But when we breed these mice, half of the mice are males. And in my heart, I could not sacrifice these mice, despite the lots of money it cost and the long latency. But we have kept a bunch of these males over time. We have data on 17 existing strains. We're currently adding 21 more to look at prostate cancer, especially with our collaborator, Ramesh Narayanan. And so to end, I'd really like to thank my lab, who are always in the animal facility, and have been really helpful. And you have to keep such careful notes on this data, especially previous members of the lab and our collaborators here in the audience. And then you'll hear at the end of this, towards the end of the session, from Jeremiah Holt, a collaborator with Neil Hayes, a physician here who's also my spouse. So thank you all very much.